recording is about to start. It says, yes, you are recording, and I'm moving over to my screen, and hopefully you will be able to see my screen, which should be this one. So can you see it? I mean the screen. Fantastic, we've got that. And you've got the topic that we're going to discuss today, protein structure. And that is one thing that I have to be very careful with protein structure with this lecture, because that is something that I learned about a lot. That was my research topic when I did lab research. Why does it say feedback on the timetable? Oh, yeah, I apologize. That is sort of a leftover. When you had to do the kiwi fruit extraction, DNA extraction practical in class, then there used to be a feedback session about what you had to write in your write-up. But since we don't do that, I've decided, together with the module convener, that we crack on with this protein structure topic. And please keep your microphones muted. Thank you. That's very good. Yeah, and the link to Moodle, Konrad has posted earlier, so please don't forget to register your attendance. We are now at a phase where we look at the attendance of students. And if we find that there is a sort of a lack of engagement, either through attendance or through Moodle clicks, then you will need to talk to your academic advisor, or they will talk to you. And please keep the microphone muted. Tamsi, please mute. Thank you. Good. Wonderful. Right. So, yeah, please don't forget to record your attendance. And apparently there was a discussion yesterday on the group chat that people want more assessments and tests. Is that correct? Is my understanding correct? Do you want more assessments? Oh. Oh, so I got the wrong. I got the wrong end of the stick. I thought you wanted that. Unofficial ones. So you mean sort of self-tests or something like that? So practice ones. Quizzes maybe. Okay. I'll see what I can do. But you are aware that for the BI 308, for the skills, there is my website where you can do loads and loads, unlimited questions. Yeah, it was only one person, but then nobody strongly disagreed. So my understanding is, if there's no disagreement, then basically this is agreement. What's the website? This is the practice calculation website. Do you want me to post a link then again and email you the link to this practice calculation website? Okay, good. I'll see what I can do. Okay, so let's get started because we've got a lot to cover today. Yes, thank you, Ed Miller. That's exactly the one. Fantastic. Guys, you rock. Wonderful. Okay, let's get started. Let's talk a little bit about protein structure. So when was that? Last time I saw you, last Friday, I think. We started with the amino acids, and I showed you that the archetype of an amino acid is basically can be written as this. We have our alpha carbon atom here, and this looks like something like that. So that is a typical amino acid. And I also showed you that under physiological conditions, the amino acids are all charged. So at the carboxy end, we have a negative charge, and at the amino end, we have a positive charge. And that is usually under cell conditions where we have a certain pH, and we will discuss pH in the BI 308 module. We will start with that soonish. But I just want to point out we have charges here, and of course we have these side chains, which are usually abbreviated by an R, which can be, in the easiest case, could be a proton. So that would be the amino acid glycine, to more complex amino acids. And I told you that you really need to know the amino acids. You don't have to draw them. I will never ask you in an exam to draw amino acids. But you need to recognize amino acids, and most importantly, you need to know the properties. So, for example, you need to know that the 3 amino acids that can be charged positively in the R group be lysine, arginine, and histidine, for example. And as people say, it is the Zwitter ion, which indicates that we have a positive and a negative charge in the molecule. We usually will... Wow. C's. You can't say that to a student, C's. We always write proteins in a direction, and this is similar to DNA. In DNA, we said we always write the DNA from 5' to 3' direction. Proteins, we write from the N, from the amino terminus, that's this one here, to C terminus. And the C terminus, that is where the carboxy group sits. And there is very clearly a huge difference. Like the DNA, proteins have the direction, and this direction is important. So, for example, the protein written in the 1-letter code and that is a perfectly valid protein, or peptide, I should say. Peptide, because it's fairly small. So here we have a lysine, leucine, they are very different, alanine, proline, alanine. Here we have the N-terminus, which is positively charged. And here we have the carboxy terminus, which is negatively charged. And if we compare that to the molecule N, positively APPALK, negatively charged, this is definitely not the same thing. Because here, for example, with the lysine, we have a positive charge in the R, in the rest. So we've got two positive charges next to each other, whereas here, in this case, we have the positive charge next to the negative charge. So these two peptides are absolutely fun, and it depends on the direction. Now, what dictates the order of the amino acids and which amino acid sits there? Well, of course, we know that this is the genetic code, which, of course, as I told you last week, it's not universal, the genetic code. There are some specific differences between certain organisms, and one needs to be careful with that. But the genetic code actually dictates what is called the primary structure of amino acids. And here the amino acids are given as the 3-letter code, and I expect a good student to know the 3 and the 1-letter code. I usually don't bother with the 3-letter code. I always go with the 1-letter code, because as a protein chemist, this is what I'm used to. So, and the question is, the Zwitter ion, is it between pH 2 and 9? Usually, yes. But we will discuss that in more detail when we talk about PAs and pKas and things like that. So don't worry too much about that. So what is called the primary structure of a protein is determined by the genetic code and the amino acids and the sequence of these amino acids. And as I told you, the sequence really depends on the orientation. Here's also something you probably, you definitely need to know as a biologist. Every protein starts its life with one specific amino acid at its N-terminus. Does anyone know what this particular amino acid is? That is sort of the start codon in the genetic code. It's the start codon, and this is M. Every protein that is synthesized by ribosomes starts with an M. But not every protein that then is in the cell does the M have at the beginning. Wait a moment. Is Klapper lying? He says every protein starts with an M, and then he says in the next sentence that not every protein, when you look at it in the cell, has an M at its N-terminus. What's happening? Weird stuff. Shemek, you're absolutely right. Proteins are modified, and it is usually the second amino acid that determines the fate of the M. Depending on the second amino acid, the M can be, how do I draw this? Can be cleaved off. So it disappears. And the second amino acid then, if we remove the M, the second amino acid becomes the first amino acid. It is cleaved off. And the second amino acid sometimes can then also be chemically modified. And everything is really dictated by the second amino acids and sometimes a little bit by the third amino acid. So every protein starts with a methionine, but this then is processed and cleaved off depending on the nature of the second amino acid. So we've got our primary structure. In order to be biological function, it has to adopt a very specific final structure. And this final structure, this is also referred to as the native structure. That's, I think, probably a good word for that would be wholesome structure. This is the structure that is biologically active. So biologically active. And depending on the protein, this can be just simply arrived by adopting what is called the tertiary structure, or if several proteins, protein chains come together and they bind extra things, for example, hemoglobin or something like that. Brilliant, Jody. I like that, the flashcards. If several protein structures come together, this could also be the quaternary structure. So in hemoglobin, we have got, for example, 4 times the same polypeptide chain, which has to be correctly folded into the native conformation for the protein to be active. So we've got the primary structure, and I abbreviate it like that. So what the heck is a secondary structure? Now, the secondary structure, and I abbreviate that with this 2 and the superscript, this 0 or something like that. What is the secondary structure? The secondary structure are local interactions mainly between the atoms of the backbone, atoms of the protein's backbone. And the backbone really is the peptide along the peptide bond. Well, I don't hope that your proteins have a bad posture, because then they won't be able to do the things correctly. So, of course, there are also exceptions to that. It could also be, so between the atoms of the protein's backbone along the peptide bond, and also interactions between the different R residues, between Rs. So what kind of interactions Hang on. I need to sneeze. I'm so sorry. I hope I didn't blow your eardrums. Thank you. Thank you very much. So, what kind of interactions do we know of? Of course, we have hydrogen bonds. Hydrogen bonds. And I give you examples for that. What else do we know in terms of molecular interactions? Oh, disulfide bridges. That is an interesting one. Yes, you are right. What kind of interaction is what is called a disulfide bridge? So the disulfide bridge is here. What kind of interaction is that? Ellie says it's a covalent bond which derives through oxidation. Absolutely right. So we can have covalent bond. That's right. It is covalent. Which amino acid would be able to form disulfide bridges? We are looking for 2 amino acids that contain sulfur in them. And people have already said it is cysteine and methionine, so C and M. Can both actually form disulfide bridges? Are both able to form disulfide bridges? Yes? What do you think? So you need to look at the structure of the respective R group for them. Only one yes. Does anyone want to upvote? No. Only methionine. Okay, let's have a look. So in cysteine, what we have, if we just look at the R group, we have an SH and we have another SH. And if we oxidize, we basically remove these 2 H's and we get SS like that. So that's the oxidation process. So that is what cysteine can do. So cysteine gives us the disulfide bond. What about methionine? In methionine, we have what group attached to the S instead of the H? We have a carbon, yes, and more specific, we have a methyl group attached. Exactly. And even with the best will in the world, we would not be able to form a disulfide bond with that. What I'm talking about at the moment is actually all the interactions that we could have in the first place. And we are then going back to the secondary structure and see which interactions are actually realized. So I just wanted to go through these different interactions that we have. What else? So we've got hydrogen bond, we have covalent bond. What else can we have? Hydrophilic and hydrophobic interactions. Now that is interesting. That is really interesting. Let's do the ionic ones first. These would be usually called electrostatic interactions. Electrostatic interactions, where you have sort of what is generally called salt bridges. Salt bridges. So you've got a positively charged residue and a negatively charged residue, and they sort of build an interaction with each other. We can also have not totally ionic, but sort of semi-charged, where we have what is called a dipole, where we have a little bit of, and I write this as delta plus and delta minus. We don't have a fully blown total charge, but sort of negative and positive semi-charges. And there can be very different forms of dipoles. And then people have said, so this is the electrostatic interaction, and they are incredibly important. And we can also have what people call the hydrophobic interactions. Hydrophobic interactions. And this is, again, another chapter where you have been lied to. I'm afraid. I hate to tell you that. Why I'm saying that. There's no such thing as a hydrophobic interaction like these other interactions, because the hydrophobic interaction just simply comes from the protein. So let's say we've got, here is our protein. And let's say we have hydrophobic residues here, here, and I just indicate them like that as hydrophobic. A salt bridge is, yeah, I think people have answered that, yes. So we have this open form with all these hydrophobic residues. These are the hydrophobic amino acids that you need to learn. And we are in environment. Now, question to you, do these hydrophobic residues like the water environment? It's an absolute no. You are absolutely right. They hate it. That's why hydrophobic. Phobos means fear. So what will happen is that the water environment that sits around will force the protein to adopt such a structure that the hydrophobic amino acids are mainly shielded inside the protein where the water has no access to it. So that is also referred, absolutely, the water is bullying the hydrophobic lads. So this is also referred to as the hydrophobic core of the protein. Whereas if you have hydrophilic residues, these will be placed on the outside of the protein. And you can immediately imagine what would happen if you put a protein into an environment that is exactly the opposite of water. What would happen to the hydrophobic core if we have exactly the opposite of water? So if we have a very hydrophobic environment, it will, absolutely right, the protein will sort of be turned inside out. So that now the hydrophobic residues in this opposite thing will be exposed because the hydrophobic residues want to be in a hydrophobic environment. And our hydrophilic amino acids will try to be buried in inside the protein so that they are shielded. So if we change the solvent, proteins very often will lose their correct structure. Because, as I said earlier, a protein has to have the correct structure in order to fulfill its function. And usually the function will be such that it is within the environment of the cell. Of course, we have proteins that are in the lipid bilayer, in the cell membrane, and there we will find that the hydrophobic residues might be outside or exposed to the environment because it is a hydrophobic environment in the lipid bilayer. But you will do that in a different module, I think. So what does the secondary structure look like? Secondary structure. So we have done the revision of the different interactions. And as Edith said earlier, secondary structure are predominantly held together by hydrogen bonds. Hydrogen bonds. And let me see if I get a nice picture of this. Where is it? Oh, yes, I think this one is the right one. Yes. Oh, hang on. No, I'll do that in a different way. I will add that here. Photo library. And that's not the one that I wanted. This one here. Yeah. Here we've got the things that can happen. We can either form what is called beta sheets, pleated sheets, or we can form an alpha helix. The alpha helix, as you can see, it's different to the DNA helix. There's nothing similar apart from the shape between DNA helix and the protein helix. Has nothing to do with each other. And we can have other folds and things like that. But these are the two major secondary protein structure that we will encounter in most proteins. So the alpha helix is between usually adjacent residues, whereas the beta sheet can be between residues that are far away from each other. But what is common to both of them is you see that here we have an interaction. We have a hydrogen bond. Let's see if I can zoom in. We've got a hydrogen bond between the amide, and that is in the backbone, and the carbonyl group of a close nearby residue. John, I think, yeah, that's pretty much how you can describe it. Curl spaghetti amino acids. Although it's probably not exactly what you should write in an exam, but you know, that is what it is. In alpha helix, we usually find in common helices, we find that is here, if we call that I, then this is I plus 4. So every fourth amino acid makes this, has this backbone interaction, and that this hydrogen bond holds the helix together. In beta sheet, the structure is again, we see here hydrogen bonds, predominantly hydrogen bonds, but they can be of very far amino acids. They don't need to be very close together, like in the alpha helix. Does that make sense? And of course, there are proteins that can be predominantly alpha helix, or they can be beta sheet, or mixture. And of course, we can combine, for example, an alpha helix with a beta sheet. And we can have, for example, another alpha helix sitting here. We can have a beta sheet that goes in that direction. Then we can have another beta sheet that goes in that direction. Here we would talk about an anti-parallel beta sheet. Or we can have a parallel beta sheet. Everything is possible. And the way this probably looks like is a little bit like that. Let me see if I find a nice example. And you can see how this protein actually starts to adopt these structures. The blue one is the N-terminus. That's usually how it is labeled. And the more red it becomes, the more it is towards the C-terminus. And you can see how this protein then actually adopts its fold. It has these two long alpha helices and probably two different beta sheets. And if you are talking about DNA, I would say we probably missed a few lessons because we are talking about proteins here. This is what the protein does. This is just a representation how it should look like. Now, the protein will adopt very quickly secondary structure. So we get the secondary structure. So secondary structure, but we then need to rearrange the protein into the tertiary structure. Tertiary structure in order to make the protein functional. The I and the I plus bit is basically, if this is, say, if this here is the 20th amino acid, then the interaction with the backbone here will be with the 24th. So the I is just a number and it indicates the step size. So that's I and I plus 4. Okay? So in order to get the functional protein, we need to fold the protein correctly. It needs to fold. Fold into correct shape. This is one of the big things that we are currently trying to unravel. How does a protein fold? What we do know is that stretches of amino acids, which are called domains, these are usually around 100 to maybe 200 amino acids. These are blocks and they can fold independently from each other. They can fold independently. And if you look at proteins, most proteins are made of one or lots of different or the same domains. And each domain can fold independently. And folding starts very quickly, even sometimes when the protein is still synthesized. So folding immediately even in synthesis. So the protein emerges from the ribosome and starts the folding process. Now, how quickly does this happen? And who tells the protein what is the correct structure? Is it just simply that the protein tries to find out what is the most stable structure? Because this is what the native structure usually is. How to find the native structure. How to find the native structure so that the protein is able to do its biological function. Let's say we have a protein of, say, 100 amino acids. This by no means is a big protein. In fact, I would say this is one of the really small proteins. And because proteins can be, anything from 50, 60 amino acids to huge molecules with thousands of amino acids in it. So if we have 100 amino acids, we can show that there are probably around, say, around 200 angles, 200 angles in peptide bonds, angles in the peptide backbone. That's just in the backbone. Okay? And let's assume that for each angle, we have 3 stable orientations of the amino acid. That would give us, for a 100 amino acid protein, the number of possible arrangements that we can do is 3 to the power of 200. Let's just simply assume that the protein is trying randomly three stable orientations in which the backbone can orient itself in this area. It's probably a total underestimate. It's probably hundreds of different stable orientations, so that it gets a little bit like origami. Yeah, you're absolutely right. So the protein is trying randomly. Let's see what this is. It's 3 times 3 to the power of 200 possible orientations. Possible orientations that the protein has to try out. Let's say for each orientation that the protein needs to try out, it takes about, what shall we say, 1 picosecond. 1 picosecond. What is a picosecond? Yeah, 10 to the power of minus 12. 10 to the power of minus 12 seconds. So tiny, tiny, tiny. Yeah, you're amazed, aren't you? So amazed that you have your microphone on. So what we can do is we can divide 3 to the power of roughly 200. We multiply that by 10 to the minus 12. And that gives us the time a protein takes to fold. Takes to fold. That's the time the protein takes to fold. If you do that calculation, it gives you something in the range of somewhere 10 to the power of years. What do you think? How many years? 10 to the power of years. Something in the range of 10 to the power of 70 to 75 years. 10 to the power of, yeah, you're right. It's 10 to the power of 77 years. So for a lousy protein of just 100 amino acids to go through every single option that it has, it would take it 10 to the power of 77 years. How long does the universe exist for? Much, much less than that. The universe exists for about 1.4 times 10 to the 10 years. So our protein would need far longer to fold than the universe exists for. But obviously the protein does fold because we are based on proteins. Lehmann's paradox. And it really shows that the folding process is not just simply trying everything out, not random tries. It can't be just random tries, because then the protein, we would all be unfolded still. So there must be some rules, which this really nicely shows. There must be rules that dictate how a protein needs to fold. Because usually folding a protein, or the protein folds within seconds to minutes. It's a very fast process. It can't be a random process. It must be guided by some rules. Now, at the moment, we don't know these rules. We are trying to figure out what these rules are, but we are not entirely clear what they are. And there's a lot of research going on about protein folding. What are the rules? What is the sort of the code for folding? And also, very importantly, if we know the rules and if we know the code, if we know that, we can figure out the structure, the native form of any protein. We need to understand that what guides the primary structure of the protein, the amino acid sequence, what dictates, what are the rules so that this protein can actually fold into its native structure. And once we know what the native structure looks like, then we can do a lot of fancy things with it. I'll tell you a little bit more about protein folding and how we are trying to figure out what is going on in the session tomorrow. Thank you very much for watching, and I see you. When am I going to see you? I'm going to see you tomorrow in our Thursday session. Have a lovely day, enjoy, and don't do anything I wouldn't do. Thank you.